I'm Matteo Di Bernardo. I'm a graduate student in the Cheeseman Lab at the Whitehead. Today is our first inaugural Innovation Initiative distinguished lecture, and we'll be hearing from James Zhao about computational biology in the age of AI agents. Before we start, I just wanted to do a quick thank yous. So Monica, I don't know if she's here or outside, has been instrumental in setting really just getting all the logistics set up and getting you guys in from the back door. And also a huge thank you to Valhalla Whitehead Fellow and Chair of the Whitehead Innovation Initiative, Alison Hamelos, who you will hear from after the lecture to introduce Rick. So this lecture is part of a larger initiative here at the Whitehead, which is called the Innovation Initiative. We're in our second year now, so we've learned a little bit about what works, what doesn't work, and heard some community feedback. And so we're very excited to kind of get into, this is a QR code about the Innovation Initiative, but get into kind of some pretty central pillars that will distinguish this going forward. So one of the things that you'll see over the course of this year is a seminar series. So part of this program is funding for various labs, and all of those labs will give a talk throughout the year about the work that they're doing through this grant. A second thing is our yearly symposium, which we had in April of last year, and we'll be doing again this March. And then we will be continuing our primer series also starting next month, which is kind of an internal discussion for Whitehead members. And our fourth pillar is kind of what brings us here today, which is the Distinguished Lecture Series. And the main purpose of this is to really increase our understanding of computational topics that relate to foundational biology. Moreover, they give us the chance to really focus in on one or two speakers and put that speaker in discussion with a Whitehead faculty member to really relate that to the foundational work that we're doing here at the Whitehead. We'll be hosting two other talks, just like this one, throughout the course of this scholastic year. So please stay tuned, and it'd be great to all have you here again. Now, importantly, an important acknowledgement, none of this would be possible without the support of Michael and Victoria Chambers. So we want to just, again, thank them for their generosity, vision, and committed partnership. That support has really created and continues to create an outsized impact across our community. Okay, closing up. My name is Matteo Di Bernardo again. I'm a computational biology student and in the Cheeseman Lab. I work on approaches to understand gene function through large-scale screens of cell morphology. And so we've really found in the Cheeseman Lab that both extracting meaningful data and then kind of interpreting that data for follow-on mechanistic experiments is really augmented by new AI approaches. So it's really, really exciting for me, and I hope for you all to have Dr. Zhao here. We're really eager to hear from experts like him about how this field can really best leverage these emerging technologies. So we'll start off today by hearing from our honored guest, and then I will turn it over to Alison, who will introduce Rick, and we'll have a 20 to 30 minute discussion session between them, followed by some QA. It's a really exciting moment for connection, and I hope that you guys all enjoy this 1.5 hour session. So with that, I'd like to invite Dr. James Zhao to the stage. He's an associate professor of biomedical data science, computer science, and electrical engineering at Stanford University, and he works on developing cutting-edge AI for biomedical applications. He's developed a number of widely used innovations, including EchoNet AI, Gradio, and SynthMol. He has received most recently the Overton Prize, which is very exciting, congratulations, as well as a Sloan Fellowship, an NSF Career Award, and 2 CZI Investigator Awards. So I'll turn it over to him, listen with curiosity, and really imagine kind of the integration of a lot of these methods into the work you do and where it can take us. Thank you, and welcoming James.
Okay, great. Well, thank you so much, Matteo and Ruth, for the introduction. It's really a pleasure to be here. I just mentioned that I used to be a student down the street. I often came here for the cafeteria and also for the lovely seminars and discussions. So really excited to be back after 10 years. So I'm a computer scientist, and a lot of work we've been doing recently is around building these AI scientist agents that can help us to accelerate and to automate some various aspects of biological discoveries. So I want to share some of the work we're doing there and also use this sort of as a motivation for some of the umbrella for conversations and discussions. So the context of this is that many people here know well that AI has been very useful in biology, and it's often used as a tool, right? When you think about AI as a tool, then often you start with a well-defined problem, right? And then you have a tool for tackling that specific problem. So for example, if you have this well-defined problem of predicting structures of proteins, then AlphaFold is a very powerful tool for tackling a problem. Or if you have a problem of like segmenting out cells, then there are other deep learning methods for doing that specific task. There's been kind of a paradigm shift over the last year in particular, where people are starting to think of AI not as a tool for just tackling individual well-defined problems, but really as sort of almost like a co-scientist that can help researchers across this broader set of research endeavors, right? Everything from helping to come up with hypotheses, questions to ask, to analyzing data, to even helping with like, writing papers and drafting and other aspects. And a lot of that development behind the co-scientists comes from these recent breakthroughs of what people call AI agents. Right? So you can essentially think of these AI agents as basically being systems that one can build on top of large language models like ChatGPT, whereby you endow these systems with the ability to use different tools. So for example, you can have ChatGPT that actually uses AlphaFold, but they can also talk to each other, talk to other AI systems, and also talk to human researchers through natural language. And these kind of systems now are quite flexible, and that's what enables it to start to tackle this broader range of research endeavors beyond solving very specific problems that can be solved by individual tools. So this topic of the AI agents that I want to explore and discuss today, and also happy to make this quite interactive. So if you have questions in the middle, you can feel free to ask. We don't have to wait until the very end. So what I want to do is to give a few illustrations of what are the kinds of AI agents that we could build, and then how do we try to use them in different applications in biology, and also some of the limitations of these AI agents, and also how that complements sort of human expertise. So the first example I want to share is the system that we built recently that we call the virtual lab. It's kind of like a replica of my physical lab at Stanford. So in the virtual lab is a team of AI scientist agents. There's an AI professor agent, and then under the professor, there's actually a bunch of different AI student agents with different expertise in immunology or computational biology, and they have their own group meetings and they work together. We give them a budget to do these different experiments. All right, so I'll explain a bit of how that virtual lab works. And then to help these agents, we also built like kind of like a replica of Stanford, so the agent school, where the agents can actually go to the school and then teach themselves how to become a better expert in one of these domains. So I will first explain how we interact and how the interaction happens with these virtual lab agents. Right, so usually we interact with these agents by talking to the AI PI. So I can explain to the AI PI some high-level description of the problem that I'm interested in, the project. For example, the project could be like, I want to analyze this very complex omic data set, or I want to design a new protein to do this kind of, to target this kind of targets. Right? And then for each project, and the PI agent would say, here are the different areas of expertise that are needed to tackle that project. And then it's the job of the PI agent then to create and to train the sub-agents that have that relevant expertise. Right? So for example, for a particular project, the PI agent might say it's useful to have a computational biologist on the team. Then the PI agent would actually automatically create and to spawn and train that sub-agent. So I'll come back to you in 2 slides of how we actually, how the training process of agent happens to the agent school. But for now, you can think of each agent as being like a kind of a GPT-4 or GPT-5 model under the hood. And then we do additional fine-tuning of these agents to enable them to better use tools and also to better be able to learn about a specific technical domain. And then once the agents are trained, right, then they can actually start to have their own group meetings. Right, so the agents can actually meet together, similar to how we meet, and they can all meet together and start to contribute ideas sequentially towards some research project, research design. They can also have one-on-one meetings where one of the agents will meet with the PI agent to maybe review a piece of code or analyze a data set to solve like a particular subtask. Right, so they can sort of alternate between these group meetings and then these one-on-one meetings. And all of those meetings sort of happen in kind of like a virtual environment, right? It's sort of like a sandbox that we created so that the agents can actually communicate and then use these different tools from computational biology efficiently. So just to make it more concrete, so here's sort of a verbatim transcript of one of these example virtual lab meetings. Right? So this is actually the first research project that we give the AI scientists to tackle, which is to basically try to design good binders to the recent COVID variants. So this was, we started this project last year, and there were a bunch of these new COVID variants. Right? So it's kind of a very timely, but also quite open-ended, challenging project. So in the beginning of the meeting, so the PI agent, right, so sort of first just lays out to the other agents what the goal is, right? So the PI agent here would say, the goal that we want to design binders, right, to the recent COVID variants, and explains why that's important. And then the different agents then can speak up one after another to contribute their ideas. Right? In this case, the immunologist agent spoke up next, and then made a kind of an interesting suggestion of designing nanobodies in, as the binders. This is already kind of perhaps surprising because often if you ask our colleagues, they might say, let's design like antibodies, which are more commonly used. And nanobodies are sort of small cousins of antibodies, and they're much less common. They're only found in a few animals like camels. This kind of maybe a little bit an unorthodox suggestion, but as we'll see, this actually turned out to be quite a wise decision made by these agents. So then the ML agent spoke up next, right, and says that, oh, I actually like this idea of designing nanobodies rather than antibodies because these nanobodies are smaller. That makes my job as the ML agent actually easier because it's easier now for me to use deep learning models to study and predict their structures. Right? And then the CompBio agent spoke up next and says that, okay, in addition to using these deep learning models, I also recommend using these more like physics-style models, right, to look at the molecular dynamics and simulations to get a more robust model of these structures. And in all of these meetings, we have like a scientific critic agent, sort of like a reviewer that sits in on all of your discussions, whose job is to try to provide more skeptical feedback to try to poke holes into the ideas of these other agents. Right? So in this case, the critic agent says that, oh, if we're going to look at nanobodies, there's not a lot of public data about nanobodies for COVID, so you need to be like super careful about overfitting your machine learning models for that task. Right? So the actual meeting goes on for longer. They have multiple rounds of discussions. So here I'm just showing you the transcript from the first part, but you can already see how the agents can interact with each other and how they kind of build ideas off each other and to reach some sort of a consensus about the research plan. So one thing that I'm quite envious of these AI scientists is that their meetings are much more efficient than my meetings. So one of their meetings maybe take a few seconds, right? So in the time that I had one of my meetings, they already had hundreds of research discussions. And because the AI scientists, they don't get tired, they actually run multiple research meetings in parallel. So basically for every question that they want to discuss, they actually run 5 meetings in parallel discussing the same question. Now in these different parallel meetings, right, so different agents can speak, and there's also some randomness behind the language models. So they often come up with different ideas in these parallel discussions. Right, so then we ask the PI agent to read the transcript of these parallel meetings to come to a consensus, right, of what to do next. Right, so this is kind of an effective way to kind of explore a broader range of ideas in parallel and then still have like a concrete set of to-dos. So this is sort of hopefully give you a flavor of how the agents interact. And in case you're curious, so here I'm showing this pie chart, basically how often the different participants would speak in these virtual lab discussions, both the group meetings and also one-on-one meetings. So like in blue here, it basically shows the human researchers, like myself, my students. So we speak about 1% of the time. Right? So our role is to mostly provide, let's say, more high-level guidance. We think of ourselves as sort of advisors to the agents. We provide high-level guidance. We tell them, here are some of the projects that are interesting, useful resources, and also what are the constraints that we have. Like, for example, how much money we have to do the experimental validations, our budget. So they can't go too crazy. But otherwise, we don't want to micromanage the AI scientists too much. So it's up to them to come up with their own research plan and execute and implement that. In this case, the CompBio agent actually does the most talking among the AI agents, which I think makes sense because the computational biology agent has a lot of work to do to basically try to come up with a plan for how to design these nanobodies and how to validate that and all the experiments and analysis. the machine learning agent's job is to actually basically do all the coding for the other agents. So it's basically like another scientist agent would tell the ML agent, implement this algorithm or develop this model for me, and the ML agent will actually do all the coding for that. Right? So it doesn't talk as much through English, but it actually does a lot of work through basically writing the code. So this is basically how the agents would interact. And any questions about how they interact? Yeah, that's a good question. I think similar to in human discussions, basically the order with which the scientists speak often affect the trajectory of the discussion. So we do see that here as well. It depends on which agent speaks second or first, right? So that also affects what ideas come up. So we could actually randomize the order of the meetings across these parallel meetings. And that's often, I think, an effective way to explore like a broader range of topics and still have the PI agent then reach a consensus across these different orders.
Can you offer like an intuition why a collection of basically different language models would be more efficient at this problem than just one language model? Because the age is based on the same model.
Yeah, so great question. So these agents here, as I'll explain the next slide, when they go to the agent school, they then start to specialize in these different topics. And we found that when you have different agents with different expertise and they have these group meetings, sometimes they get into debates and disagreements. And when the AI agents have disagreements, they're very polite about it more than humans, but they still have to resolve that somehow. And often you get into, I think, more interesting and more creative insights and reasoning when the agents have to disagree and have to convince each other. Whereas if you have a single agent to do the problem by itself, it just doesn't have all of that discussions and debates. That ends up being less creative and less robust. So we did that comparison. Great. Oh, yeah.
Group size?
Yeah, so we did some experiments where we say, okay, so what's the optimal size of the science team? And we found that if you actually have too many AI agents in these meetings, then there ends up being like too much distractions, and that also made the result worse. So the sweet spot for this project ends up being somewhere around 3 to 6 agents.
How long does it take to train the different agents on the particular experiences? And what is your criteria to say, okay, you achieve this level of experience, you're an agent?
Perfect. Now, so that's maybe a good segue. So let me explain then how the training process happened. We do this mechanism that we built of an agent school. And the motivation for this is that if you take the off-the-shelf models like GPT-4, GPT-5, they're quite good at broad general knowledge. But if you go really deep into your favorite research area, then often the model do not have the latest or the most cutting-edge knowledge or the ability in that domain. So to address that gap, that's why we build a school whereby the agents can actually go to the school and teach themselves how to become maybe a better expert into that domain. So the way that we set up the school to work is that first we tell the agents, here are some of the topics that you should learn about. Right? Maybe one topic could be like learn about the most recent developments in the latest COVID variants, right? Or learn about the latest developments in this particular tool like AlphaFold or other domains. Then for each of those topics, then agents will basically design their own learning curriculum by coming up with a set of questions that are relevant for that topic. For each question, we allow the agents to basically go out and then find relevant literature, and they can basically, for example, do a PubMed search and find open access papers and download those papers, or they can find different code bases. And then they would download those locally and then read that to update their knowledge. Right, so as they are reading these papers, right, so they're basically updating their model parameters. So what happens here under the hood is that each of these models, it's a language model, so it's a transformer neural network. And then as the agents, maybe they all start off as being, let's say, the same neural network in the beginning, like the GPT-4 neural network. But as the agents actually read these new papers, then they can actually update the parameters of this neural network to ingest that new information. So that's why, like let's say, the immunologist agent and the cell biology agent starts to diverge in their expertise because their model parameters also starts to diverge. And to your point, it won't be a very good school without some sort of assessment mechanism. So after they go to the school, we basically have like another agent to design quizzes to assess how well did they learn that topic. Right? But the quizzes can be designed from like reference materials and textbooks and papers. And then if the agents do not do very well on those quizzes, then they can't graduate. They have to go back to school, do additional readings, additional fine-tunings, right, before they can do well on the quiz, and then they can graduate and join the virtual lab. So the school part here takes about a couple of days, right, for them to start to finish for fine-tuning and finding the literature and updating the model parameters and passing the quizzes.
We were just talking the other day about how some of the open source literature is like not very high quality, and so are the agents getting a good education, and how do you write a quiz that actually assesses? How do you learn the quiz?
So that's a great question. Even for the human experts, we have to sort of figure out what papers, if they're conflicting information, how do you evaluate that, and what's more. So I think that's still like an open challenge for the agents. All right, so here, we can give it access to, we can tell it, here are some of the databases that ends up being more popular as used, right? But otherwise, we don't provide, we don't tell the agents, you have to read this or you have to do that, right? So there's they find that they do a PubMed search or a Google Scholar search and find maybe they start often with these review papers and they read those, and then based on that they find other papers. So it's sort of similar to, let's say, how a PhD student might try to go about navigating this space. So that does lead to some of the biases and mistakes which the agents can make, right? And that's where we found that often it's still useful to have this kind of some human feedback in different parts of the process, right? So it's not entirely autonomous, so that we still provide feedback to the agents, so that if they go down the wrong path, we tell them, oh, maybe you should look at this kind of tools, right, or read more about this field.
Hallucination is really a big problem in training the LLM, even with a given sophisticated evidence or publication. I'm just wondering, have you observed any type of behavior as such in agent school, where agent might be making up things, and then they start talking to each other, and really what they are learning isn't exactly what they should be learning? I'm just wondering, have you any thought on that?
Yeah, that's also a great question. So these hallucinations are still a challenge. A couple of things we found to be useful. So one, I think actually having this critic agent who's designed to be more skeptical is quite useful in reducing some of these hallucinations. The second is that by having these parallel meetings and taking the consensus often is more robust than the individual meetings. But maybe the most useful thing that we found is that we actually basically try to encourage the agents to not do too much talking, but actually try to do most of their work through coding and through using different tools. Because these models are actually much better at writing code, right? And it's also easier to debug and to evaluate this code, whereas if a scientist agent just does a lot of talking, then somehow it's more subjective and it's harder to do, right? So they actually basically have to show their work by demonstrating their work by writing a lot of code. And that actually motivates, it's illustrated through this example, the case study, where we asked them to basically design binders, and they said, okay, let's design nanobodies. And because there's not a lot of data for nanobodies, agents decided to basically come up with a computational pipeline to help them to model and to optimize these nanobodies in silico. And as I think kind of instructive to see what did they try to do, right? So here's the pipeline that they created. So they basically decided to use a bunch of quite well-established tools and combining them to help them to do this project, right? So first they decided to use this protein language model called ESM. Basically what ESM does is that it takes its input, some amino acid sequence, and it computes a score to see how likely is this to be like a functional stable protein, right? So this would be like almost like a first filter for the agents to look for the candidates, right? And then we'll pass those candidates into AlphaFold together with the spike of the target protein, right? And then AlphaFold will generate this three-dimensional structure. The agent will look at that three-dimensional structure and then pass that into Rosetta, which does more like a physics-y style dynamics to relax that and then estimate the binding affinity. So the agents actually decide to use these different tools because each tool provides somewhat complementary information about these structures, right? And then the agents would actually combine the output of these different tools holistically into like a scoring mechanism to tell it which variants to, which nanobodies to prioritize, right? It would actually iterate through this process multiple rounds to reduce the search space to come up with like a final list of candidates, right? So they came up with this pipeline, and then they would do their implementations to implement this pipeline. They also can modify the tools, right? So they modified, for example, this ESM a little bit to change the scoring mechanisms to adapt it to this problem. So I think that sort of maybe exhibits the kinds of things that agents are probably better at, right? So they're not great at, if they want to try to solve the problem entirely from scratch or come up with an entirely new method, right? But I think what they're better at is to these kind of, let's say, combinatorial creativity of taking different existing tools, right, and combining them in perhaps, and modifying them in perhaps in sort of new ways and adapting that to the project, right, and try to make that more systematic and more efficient. Yeah.
Did the user tell the agent that these are available tools, or did it suggest these tools?
Yeah, it's a good question. So we asked the agent. So which tools do you want to use? Right, so the agent then says, here are the tools I want to use as a part of one of the group meetings. And then basically then we provide those tools locally to the agents in their virtual environment so they can run AlphaFold locally in their sandbox.
A comment and question. The comment is about hallucination. It seems there is a recent paper claiming they found one of the reasons why models hallucinate because when they train the model, they didn't teach the models to say, I don't know, kind of force them to always answer something. I wonder if you or someone have tried, in the back end of the software or the platform to use prompt engineering to tell, if you don't find force it to say I don't know, if that will help us minimize hallucination. That's the question is about context window. I imagine with multiple agents, the context window will be very long over time. And when the window is too long, the model tend to ignore information in the middle or increase the error rates. And how do you manage that?
Yeah, so it's a good question. So basically, we built for each agent like a memory mechanism. So essentially, each agent have their own sort of like a lab notebook. So they remember what did they talk about in the previous group meetings and one-on-one meetings. Right? And we also have the PI agent to periodically update the entire group and say, here's everything that's we've done so far, and here are the next steps. That sort of level sets of the different agents. But you're right, having like this memory mechanism that's more structured helps each of the agents to basically stay on track. So in this case, so the computational part here, that's all down and implemented and designed by the agents. Right, but they don't do their own wet lab experiments yet. So this is where it's also critical to have this human-AI collaboration. So basically, the AI agent said, okay, so here's this list of 92 nanobodies that it thinks are maybe the most promising candidates, right? So they gave us those designs. And then we, with our experimental collaborators at the CZ BioHub, then we could synthesize these nanobodies and then test them in the wet lab, in the real world, to see how well do they actually work, right? To provide sort of the experimental validation for the virtual lab. So here are basically two example nanobodies designed by the AI scientists, and I circled some specific residues where the agents actually sort of introduced variants compared to any of the existing nanobodies. Right? So these are sort of the new mutations introduced by the agents. And the bottom here basically shows the curves from different ELISA experiments that basically show how well do these nanobodies actually bind in the wet lab to the different target spike proteins. So for example, if we look at this bottom red curve, right, with the open red circles, that's where if we take the existing human-designed nanobodies and test them against this recent JN1 variant, which started to spread last year, then that does not show very good binding. Then the second curve was the solid red circles. Is basically now if we take the nanobody that's been optimized and designed by the virtual lab by the agents, it starts to show more moderate, a little bit more promising bindings. And then the two blue curves are basically if you take the same nanobodies, right, and test them against very different strain of the virus, the one from Wuhan from 5 years ago, then the top one's designed by the virtual lab that also shows perhaps more like some enhanced binding to this very different strain of the virus, which is actually a pretty challenging problem to design, like a good binder that can bind well even across like sort of 5 years of evolution of the virus. And what's not shown here is that we did other experiments to test these nanobodies against other viruses, and they also against the host protein, and they do not bind to other viruses. So it's actually specific to the SARS-CoV-2 family.
Is this a constraint that you gave to the agents, that it should bind the original Wuhan strain as well as some other target strain? jam?
Yeah, so we didn't give that as an explicit constraint. So the agents, I think through their literature search, they certainly would know about these, the previous viruses and the previous families.
I'm sure you've heard this question before, but if the agents perhaps came up with something that's harmful, obviously you guys test this experimentally, but have you thought of controls or things that you would put into the AI stage to try to guide it towards actually developing useful things as opposed to somehow insidiously producing something that could be harmful?
Yeah, I think so. That's also, I think, a really important question and worth a longer discussion on, right? I think there are different stages of guardrails where it's useful to put in. Certainly on the the virtual to the real interface side, right? So I think it's reasonable to have a guardrail in the place where these nanobodies or any of these biologics are getting synthesized. I think that's the important place to do the checks. On the virtual side, then the agents, they do have some guardrails as well. That's actually one of the roles of the critic agents to basically review the work done by all the other agents to make sure that there are no unintended or mistakes or sort of potentially dangerous things. But of course, the human oversight is still important.
Are these agents able to integrate more complex biological elements within these problems? For instance, the difference between binding and neutralizing for a given antibody in the context of viruses?
Yeah, I think it's an interesting point. So I think the agents could integrate and try to solve even more complex problems. Actually, one of the related things we've done is that now that we have these experimental results, we can actually give this back to the agents, right? We can give these plots back to the virtual lab and then ask them to use that feedback, right? So they have actually modified their pipelines to further optimize it based on these experimental feedback. And maybe that also relates to some of these broader discussion points, right? So we actually made the virtual lab sort of all open source, right? So it could be flexibly used in other applications. It's a part of this recent paper. And it's not really limited to, let's say, design proteins, right? So because of the school mechanism, which is actually quite general, we envision these AI scientists actually could tackle quite different kinds of biomedical problems and maybe even problems in other domains like chemistry or physics, right? So I'd be very curious also to follow up and to hear from folks here, maybe some of the potential areas that might be interesting to you. And similar to one of the earlier questions, we did find that when we have teams of multiple agents that work together, right, that ends up often being more effective than having a single agent try to solve the problem by itself. I think that maybe also mirrors some of the experiences that we have when we have collaborators, human collaborators that come from different expertise. And as we said, I think it's useful to have basically equip the agents with some resources, right? So one important resource is actually a memory mechanism, like a lab notebook, so they can remember from previous meetings, but also having this virtual sandbox so that makes it safe for them to execute these tools locally. That also relates to the security question because they're executing these tools in the sandbox, then there's less risk for that to go wild. One thing I think that's really quite interesting that I'm fascinated by is that the agents also have their own social dynamics, right? So similar to in human meetings, some people talk more than others, and some people have louder voices, and that also affects the kinds of science. We also find that in these AI scientist meetings, so some agents talk more than others, and that also affects the outcome. But one thing that's much easier to do as the AI scientists that we can't really do as humans is that we can do different oblations, right? We can actually have parallel meetings where we say, okay, why don't you talk more, or why don't you stop talking, right? And do these oblations, and then see how does that actually systematically change the outcome of the results. Right? And we can also do the experiment that Ruth mentioned of what if we change the number of scientists, right? If you have too many scientists, does that actually make it better or worse? So I think it's kind of also an interesting petri dish, right, to do a lot of these, let's say, science of science experiments so we can figure out what is the best combination. We've also been doing experiments on basically putting different personalities to these agents. For example, it's actually turned out to be somewhat effective if you make the critic a bit more neurotic, kind of like a George Costanza type character from Seinfeld, right? But you don't want every scientist to be neurotic, so there's some sweet spot of having a combination of personalities that also leads to maybe more creative and more effective science. So I think there are a lot of interesting things that we can do on that front as well. Great. So in the second example I want to mention is that a follow-up work that we've been thinking about specifically of trying to build some sort of data scientist agents, right? And teams of data scientists that can help us to really get more insights out of these very complex data sets that we're all collecting, right? So the idea of this data scientist agent is that you take as input some data sets that could be like a genomics data sets that you'll collect, and the agent will actually generate a set of hypotheses and analyze each of these hypotheses in the form of these in-depth Jupyter notebooks, right? And in the end, they will sort of summarize their insights and findings into like a report with figures and statistics to summarize what did they learn. And the reason why I think data science is actually a good, particularly good application for AI agents is that the first is that, and people here know very well, is that now you have dealing with increasingly large and more complex data sets, right? And there's also just a bewildering array of different tools that one could use, right? Just even in single cell analysis now, there's thousands of tools and more ones coming out. It's often not clear to us as the end users which tools should I use to analyze this data sets. Often we only know about a couple of things we can do, or we have limited time, right? So as a result that, I think even among the existing data sets, we might only really be scratching the surface of what could be learned, right? There's a lot of insights that we might have missed even among data sets that we have painstakingly already collected. And this motivates us to say, can we basically have these AI data scientist agents that can just reanalyze even existing data, but perhaps in greater depth, right? And spend more time on this, and can they actually find new insights that we have missed, right? So the input into this agent then would basically just be the data set or any description of what we've previously done, right? And the output will be a report with the figures and also the reproducible code to generate those results, right? This can be applied to different kinds of biological data. So we call this CellVoyager agent. So very briefly, how CellVoyager works is that it takes a look at this data that you have collected. So it first does a bunch of explorations to figure out what is in the data, and then it does like a deep research to read about all the papers that are relevant for that data set to see what are the kind of hypotheses and biological questions that people have asked about similar data sets before, right? And then it also builds up the environment, like initializes and imports the relevant tools. And then it gets into this research phase or analysis phase, right? Then for each of these analysis plans, it actually starts to implement analysis, right, in code, execute that code, right, and then interpret the result and then iterate through that process. So here's like what an output will look like. So it does everything through a Jupyter notebook, so it's easy for us to interpret and also to reproduce the code by just rerunning the notebook, right? So in each of these notebooks, we first say, okay, here's what is the like a description of what is the test or analysis it wants to do, right? And then we'll generate the code to implement that analysis. It will execute that code to produce the figures, and then we'll look at the figures and then to interpret those figures in the biological context, and then iterate on that to go more and more deeply into the outcome, right? But the one thing that's the agents can do well is that they can actually do many of these, right, hundreds of these analyses in parallel, whereas maybe it will take me a while just to do one of these analyses, right, if I write my own notebook. And the way that we validate this agent and to demonstrate how it might be useful is to basically we'll take previous papers that people have recently published, we'll give those papers and data sets to the agent and ask it to basically reanalyze that data set to come up with new insights that might have been missed by the original authors. And to validate that, we just basically give the output from the agent back to the original authors to have them double check first that the analysis is done correctly, and then second that the insights are actually something that they have actually missed in the original first pass analysis in the original publications. So we did a bunch of different case studies of this flavor. I'll just show you one example. It came from sort of a nice brain aging study done by one of my colleagues at Stanford. So in the original study, they basically sequenced mouse brains across different ages. They generated single-cell RNA sequencing data. The goal of the original paper is to basically build these biological age clocks, right, to predict from the transcriptome into the biological age of different individual cells. In this case, CellVoyager actually conducted 5 new analyses. Right. The original authors found basically 4 of the 5 new analyses to be particularly interesting. So all 5 were done correctly, but 4 of these 5 are particularly interesting to the original authors of this single-cell study. So here's sort of the example outputs from one of these analyses. Right. So in this case, it's a single-cell data set. So the agent will just do some of the standard visualizations to figure out what is in this data. And then pretty quickly, so these are figures that are generated by the agents, and pretty quickly, it actually realized that there's actually a pretty interesting difference and consistent difference between what's called transcriptional entropy and the biological age, right? So essentially, the cells that are biologically older tends to have this higher entropy, right? And then it found this pattern is pretty consistent with quite robust statistics. So this was actually an interesting analysis that was that the authors thought was pretty interesting, but something that they had not thought of before in their data sets. This is not an entirely surprising finding because people have reported similar kind of trends and correlations between transcriptional entropy and age in other tissues, but not in this particular part of the brain, right? So this is kind of an example of the type of things that this data scientist agent could do, right? It can basically find, maybe some of these insights that have been missed by the original authors, but then can be validated through literature. And in this case, actually, the agent actually went and found other data sets from other studies to further validate this finding. Right. So I think there are a lot of similar kind of use cases, especially when we're dealing with with these more complex data sets. Then maybe having the agents to help us to reanalyze it or even to reproduce some of our initial analysis as a way to first make it more robust, but also potentially fill in many of the missing gaps that we might have. Yeah.
Do you feel like, for example, like are there certain like fields or papers that the teams are better at reproducing than others? Like are there certain directions in science where like it seems like the teams don't perform as highly?
Yeah, I think some of that, it's a good question. Some of that depends on how well annotated and organized the data is. So for example, if like one reason why the agents often make mistakes is because if there's not enough metadata or context associated with the data. Like say if you give it a data set, but you don't tell the agents how the data was pre-processed, right? Then the agents might make assumptions that are not valid. Right, so if you have a data set that's having some standard structures with the metadata and the method section in the paper is well written, that helps the agents quite a bit in reproducing the results.
I see CellVoyager is tuned to single-cell RNA-seq, so mRNA profiles, and I'm curious in what way is it tuned to those kinds of data sets?
Yeah, so it's a good question. Yeah, so we initially started with the single-cell use case because there's a lot of existing good tools that will help the agents can actually benefit by using some of the more standard tools. And now we're sort of expanding the framework, so it's not really limited to transcriptomic analysis, but we can also do other kinds of omic analysis. Yeah.
But I understand the input is the data set plus an existing paper. Is it possible to run it without the existing paper?
Yes, yeah, you could just give it like a de novo data set and then ask the agent to say, okay, analyze this, explore this in depth, and what do you find? In our case, we give it also like a paper and say, here's what people have done previously, and now why don't you try to find something new, right, that's not in the previous analysis, but you can use it in both modes.
So it looks like you're using maybe ScanPy or a popular single-cell analysis tool, so I guess there's probably hundreds of thousands of lines of code that have been written in ScanPy. Have you done like an ablation study where you just give it like maybe the author's documentation of ScanPy or just the tutorials and see how well it can run ScanPy, just to get a sense of like when somebody develops a new tool, how much examples of it does something like this need to see in order to use that tool effectively?
Yeah, so that's a great point. That's actually a great segue into my next part, but briefly to answer that is that I think one reason why the agents are especially adept at using tools like ScanPy is that there's actually been a lot of papers that used it, so the agents have learned from that, and it's also relatively well documented. So if we give like a brand new tool that's not so well documented, then agents do not, the language model themselves actually do not know how to use that tool, which actually motivates the last part of our mission, something new and I'm quite excited about, this idea of what we call paper to agent. Right? Essentially, the idea is to basically turn research papers, right, from these passive artifacts of research, but into active agents. Right? And the motivation for this is precisely related to the previous question, is that a lot of time the people publish these research papers, which are very passive, right? So the paper produces some code or some data sets. It's actually often pretty challenging for the end user to figure out how to use that code or how to use that data set effectively. Right? And we thought, okay, so what if we turn these research papers into an active agent. Right? So you can think of the agent sort of like being like a AI corresponding author that sort of knows how the data sets or how the paper works, right, and can basically help the end user, the readers, to actively use this tool or use the paper to apply that to their own problems. Right? So in this case, right, so instead of just reading the paper, you can sort of ask the agent, right, so, can I apply this paper methods to this new problem that I have, right? Then the paper agent will be able to interactively do that analysis for the user, right, automatically, and then generate some end results. Right, so this was done for this recent paper called Alpha Genomes, where we created this Alpha Genome agent. And briefly, how this paper-to-agent works is that essentially the challenge is that if you give a paper or give the code base to a language model, often the language model may not know how to use that code base or data set from the paper properly, especially if it's like a new paper. Right? So to address that, essentially we have this automated pipeline that we created to convert each paper into what we call MCP, or Model Context Protocol. Right? So the way this works is that we have a team of different sub-agents who would read the paper and also look at the code base from the paper. And from that, they will extract the environment that it takes to run that code, also extract different tools from the original paper. Right? And then the agents also define a bunch of different unit tests to test how well can they actually execute those tools. Right? Based on all of that, then they create this kind of MCP server. So MCP is just recently emerged sort of in this industry standard way for AI language models to interact with external resources. So in this case, it basically turns the entire paper into an MCP server that's hosted remotely. And this MCP server then would have different tools, right? Different resources, which could be the data sets underlying the paper, and also the different workflows that's learned from the paper itself. So then we can basically expose that MCP server, this remote server, to any downstream language model like Claude or Chat or GPT, and then that downstream chatbot then will become like an expert at using that tool, an expert of that paper. So going back to this point of ScanPy, right? So we can apply this paper to agent to automatically turn ScanPy into actually a ScanPy agent now, right? ScanPy agent, then we actually know about all the different modules and tools within ScanPy. So you can just ask it, okay, can you actually apply ScanPy to this new data set? And the agent will actually be able to run that and generate the results. And in this case, we can show that the ScanPy agent is able to produce analysis that exactly matches how human experts, if they try to use ScanPy to analyze the same data sets, they will get the exact same results. Another example is this recent Alpha Genome paper, which is Alpha Genome, which recently put out by Google DeepMind. It's a very nice model for basically interpreting different functional effects of different variants. Right? But it's actually also pretty complicated. Right? So then with paper to agent, we can turn this Alpha Genome paper into an active agent. So then you can just directly, you don't have to download anything yourself anymore. You can just directly ask this Alpha Genome agent. For example, in this case, we asked it, okay, can you use this to interpret this particular variant and try to find what are the causal genes, right, for LDL cholesterol. Then this agent then would come up with this workflow that uses Alpha Genome to generate these different interpretation figures, and then look at the literature, and then in the end come up with a summary of a set of findings that says that, oh, in this case, maybe this sort 1 gene is the most likely causal gene. Right? So that actually simplifies a lot of the workflow for the readers, and everything here from the agent is reproducible. So if you want, you can look under the hood and see what is the different steps in the code that's written by the agent. But for users that are that just want to actually use this tool or use the insights from the paper directly without having to download everything and figure out how to use it themselves, then the agent becomes sort of like an expert in helping the user to do that. So I guess our vision here is to sort of reimagine what these units of scientific communication will look like, right? So instead of having, or in addition to having these sort of papers, which are sort of traditional artifacts of research, but are rather passive, right? We imagine that essentially every paper in the future would actually have an agent associated with that paper, and the agent would sort of be like the experts that will be available 24/7 to help any of the readers in using the data or the code or the insights from that paper to new problems. right? So instead of having to maybe email the corresponding authors from the paper and wait for weeks to see if they reply, now you actually have this expert agent that can help you, whose job is to actually help you to do that. So we think this would be a kind of a nice way of sort of disseminating knowledge that's more accessible. And you can also imagine now that we have more and more of these different paper agents, so those different paper agents can themselves start to collaborate. Right? So maybe you have a paper agent for a new methods paper, and then an agent for like a new data resource paper, right? Then those two agents can actually get together and say, how can you apply this new method to this new data, right? And then they actually spark a lot more connections and maybe more insights. And I'll just wrap up by actually showing that we are, we're also putting all these different pieces together, and we're running this experimental conference that folks here might be interested in. So it's called Agents for Science. It's sort of the first conference where both the authors and also the reviewers are AI agents, right? So currently, most of the journals actually do not allow AI as a co-author, right? So we flipped this around and said, okay, so you have to actually be an AI to submit to this conference, right? The humans can be co-authors, but the AI should be the first author, so they should be doing the work, and the humans can be the supervisors. And then also the reviews will be done in the first round by AI agents, and in the second round, then we'll have human experts to help to do the final adjudications. All right, so all the papers and all the submissions and reviews will be publicly available as a resource for the community. I think it would be a good way for us to learn about like, what are the strengths and also limitations of AI, both for doing research, but also can they find mistakes in these papers? How good are they at reviewing these papers? This is kind of an experiment that will be sort of open. Everything will be open, and it's running now, and then we'll have these results come out in the fall this year. So there's still time if you're interested in having your agent to put together a paper and submit to the conference. Great. So I'll stop here and just say that it's still quite early stages, but we're also very excited about the potential of building these AI co-scientists to really help us across the different aspects of research endeavor. Right? With the virtual lab, we've seen how teams of different agents can work together and try to tackle some quite challenging open-ended projects that we can experimentally validate. With CellVoyager, we have these data scientist agent that can sort of reanalyze complex data and often find insights that we have missed ourselves in the initial analysis. And then the paper to agent, I think is sort of how we envision reimagine how papers should be written in the future, where every paper should have a corresponding agent that can help to expose the insights from that work to make it into, to reduce the barrier to access. Right. I want to especially acknowledge my students that led the different projects, so Kyle Swanson for Virtual Lab, Sam Albert for CellVoyager, and Jiaqin Mao for the paper to agent. So happy to take any further questions.
What is your thought about the approach of virtual labs, paper to agent, will have an influence or impact in the future scientific research? So things like about paper to agent to help to understand the paper quicker and easier, and then sometimes people do feel the process of try and fail of reading those papers is actually a journey itself. And I'm wondering, will you then have you in the future will have an agent to paper, asking the agent to start writing the paper itself? I guess I'm just trying to understand where do you see this type of approach will end up in the future, say 2 or 3 years time.
Great. Yeah, thanks for both are great questions. So for the first one, the virtual lab platform is actually, so it's open source, it's compatible with different base language models. In the nanobody experiment, there we use GPT-4o as the base model, and then we do the fine-tuning on top of that. But we also done experiments with other language models, including open source models, so people can plug in different models to apply that to different tasks. You can use like GPT-5 or Anthropic models. And the way that we think of these paper-to-agent is that certainly there's a lot of value in really deeply understanding the paper ourselves. But there's also quite a lot of technical work in just getting the artifacts of that paper to really apply that to my own problem, right? So if the paper has like some code base, if it's not super well documented, often it will actually take a student or take a researcher quite a bit of time to figure out, does that actually work? Sometimes the code doesn't work, right? And it could really waste a lot of time. And also requires a level of technical expertise to do that and run that locally. It's maybe not, could be a barrier for a lot of people. Right? And our goal with the agent then is to say, okay, the agent is basically going to be like the expert assistant who could simplify all of that process, so then the user can just directly have the agent, ask the agent to basically do then, to use that tool or to apply that tool to their new problem. You can even ask the agent, is this tool from your paper applicable to my setting, right? And maybe that could really accelerate the process quite a bit.
When do you guys reinitialize your models? Or I guess for your weights, do you have stopping criteria or restart criteria for any of these projects?
Yeah, so I think it's a good question. Some of it depends on sort of the memory aspects of the models, right? Essentially, here, if we have the agent start on a new project, so by default, we don't give them the memory from their previous projects unless it's very related. But I think that's sort of an interesting point is that a lot of the human scientists, like we learn actually from doing a lot of previous projects, and there's sort of both the mistakes, the success, and the failures. And I think there's actually some value for this, maybe even having longer term memory for the agent, so they can actually learn from all these other projects, even if it's not directly relevant, but they can learn about sort of the process of doing better science.
Scientists input into these different technologies, is that used in any way for retraining, or how does the confidentiality or intellectual property kind of would look like for that?
Yeah, so that's also a good question. So here, for example, like when we have like the paper to agent, then it's just taking everything that's publicly available as released in the paper itself. Right, so if the paper releases the data, then the paper to agent then will be able to convert that data into the MCP server. But if the paper does not release that data, or if it's like PHI, then the agent does not have access to that.
Of paper to agent, like for example, some research code is not error-free. Will it be able to like catch those errors and then just fix it by itself?
So that's a great point. I think it works more reliably if the code is well documented and if also the paper itself provides enough information. I view that almost, maybe you can even view the paper to agent sort of almost like a metric itself, right? If a particular paper is so poorly written and the code is so poorly organized that cannot be agentified, that probably is also a signal back to the original authors that they need to provide more information in the methods section of their paper. Right, or make the code better organized.
We have a question back here, and then we'll do one or two more, and then we'll shift over to Allison.
Hi, I guess correct me if I'm wrong here, but my understanding of how you fine-tuned the virtual lab was you fine-tuned agents specifically with AlphaFold multimer and nanobodies and ESM. And so I guess like why is it surprising that it came up with the pipeline that it did?
Yeah, so just to clarify, so the fine-tuning process is basically having the agent, for example, learn from the broader literature, right? So for example, we actually downloaded a bunch of paper about the recent COVID variants. Right? So not specific for AlphaFold or other things, but just try to learn about like what are the different variants and what are like the mutations in those variants, right? So here it's not like specifically trying to being fine-tuned to just this subset of tools, but learning from the broader literature.
Hi, yeah, that was a really cool vision for what science is going to look like in a few years. What do you think are the gaps for, that are really worth human trainees, postdocs, investing their time and energy in doing beyond building agent frameworks for science?
Yeah, so it's a good question, and I didn't have time to go into this part, but I think the way I think the agents, the current limitation why humans are very complementary is that the current agents, at least for now, are still better at putting together existing tools right, in this kind of combinatorial creativity of putting together existing tools into like new pipelines, right? But they're not great at really developing an entirely new tool from scratch. Right? And I think that's actually complements a lot of what people here are doing, right? Really thinking about how do you build like the next great new technology, either on the experimental side or on the computational side, right? Or the next AlphaFold, right? So I view the agent and like the human experts ideally would really do this cutting edge, more in-depth work and building the new methods. And the agent then will be the interface of exposing and combining these methods and applying that to a broader range of problems.
I think we're going to shift to the next section, but we'll also open it up to questions afterwards.
All right, thanks so much, Matteo. All right, everybody. So thank you so much for having more questions. As Matteo said, there will be time for those soon. First, I want to thank so much Dr. Zhou for his incredible talk. I think I can say without exaggeration, it was unforgettable. I think we will not forget this anytime soon. I know we're all really excited to dive into deeper discussions with him. So perhaps we could start with a round of applause for Dr. Zhou. Thank you so much. All right, so my name is Allison Hamelos. Some of you know me, some of you may not know me. I'm a Whitehead Fellow here at the Whitehead Institute. I'm also chair of the Whitehead Innovation Initiative Community Leaders Program. It's a long title, but essentially I help facilitate folks in the community who are excited and passionate about the vision of melding AI and machine learning with biomedical innovation, with creating amazing events like today. And along those lines, I think we owe a huge thank you to Matteo Di Bernardo for his incredible work putting together this entire event. It's really impressive as a grad student balancing his research as well as excitement about the initiative. So maybe we could give a round of applause for Matteo. Really well done. As well as Monica Concepcion, who also planned this amazing event for us with Matteo. So thank you to Monica as well. Thank you so much. All right, so if you are interested in being a part of events like these, please do reach out to us. We would love to hear from you. And we have many, many more amazing events planned for you throughout the year. This is just the first one, although I have to say I think it started off very well. All right, so now we have to transition to introduce our next big thing here, which is going to be a conversation between Dr. Rick Young and Dr. James Zhou. So I will introduce Rick, since we already know Dr. Zhou. So Rick is a longstanding member of the Institute. Oh, that's interesting. Maybe I should say this first. Rick is a longstanding member of the Whitehead Institute. He's been here for a long time and has made a huge impact here at the Whitehead. He's personally quite an inspiration for me. He is also a longstanding member of the Scientific American. Okay, here we go. I see what's happening. He's a longstanding member of the Institute, and the Scientific American has recognized him as one of the top 50 leaders in science, and I think that's well justified in technology and business. He's also been the recipient of many awards, which I probably cannot list them all, but I will note a few. The Burroughs Wellcome Scholarship, the Chiron Corporation Biotechnology Research Award, and Yale's Wilbur Cross Medal. He's also served as an advisor to Science Magazine, the National Institutes of Health, and to the World Health Organization. So Rick is also a member of the National Academy of Sciences and the National Academy of Medicine. His lab is integrating lab-in-the-loop AI into their work with biomolecular condensates, which I think is an exciting complement to the work that Dr. Zhou was telling us about, combining research with AI-driven discovery. So I think this is the experimental validation side of things. Rick's lab really combines these two things quite well, and I'm very excited to hear the discussion between these two pioneers. So from there, Rick, Dr. Young, and Dr. Zhou, we are excited to hear your conversation. So, thank you.
James, first of all, welcome back. It's nice to see you here, and it's amazingly fun for me to see the level of creativity you're injecting into this area. And first of all, I just want to ask you, are you having a lot of fun?
I am. And one thing that I should mention, I mentioned to Rick, is that So Rick actually taught me everything I know about gene expression, gene regulation. I took his class when I was a student next door, and so that's especially fun to come back here to MIT and to the Whitehead.
Yeah, well, in that class, I usually tell students that, I usually explain to students the impact that they'll make in the future, but here's a great example of the impact. So I want to ask you, I want to cover a number of topics where I think you can help this group think a little bit more about this space. And one is rate of discovery at this scale. We're seeing accelerated rates of discovery that were unimaginable just a year or two ago. And there's a kind of impedance mismatch between the rate at which effective compute is increasing and the rate at which we actually do experiments to explore it. But given that, what areas of biomedical science do you think are the most likely to see real paradigm-shifting breakthroughs from AI in the next 5 years? This is what you should be working on.
Yeah, so I think the rate of discovery has changed and increased quite a bit. And I asked my own PhD students, what is their own assessment? Like, what is their productivity now compared to 2 years ago? Right? Because of these AI tools, and I think the median report, self-report, was like around 2x. Right? People just feel like they're much faster because of support with coding from these agents, the ability to do literature review much more quickly. And I think that has both challenges and also opportunities. I think one challenge is that there's also just even more noise out now, right? Just because now it's easier to put things out and easier to do these analysis and easier to have these reports. And that somehow also raises the floor, right? Like you have to now be even more innovative and go deeper into a particular field and then do the experimental validations and to really be careful to really stand out among that rising floor. I just come from more and more papers coming out from these AI human efforts. And in terms of the opportunity side, I think probably one area is that often the bottleneck for the human as a researcher is that we have fairly limited context, right? I was mentioning to Ian before that I can probably keep track of like a few pathways in my head and a few feedback loops. Right, and maybe I will read like 2 or 3 papers in a good week in depth, and I can only remember a few things, right? But one of the things with the AI is potentially just having much greater context, right? So they can now read across many different adjacent fields, right? And actually have quite deep expertise with these training mechanisms, quite deep expertise in these different adjacent fields, and they're able to make a lot of these connections, right? Take an idea from field A, apply that to a problem in field B, that often I think leads to some quite exciting insights.
One of the issues I have is data quality and bias, and I'm wondering how you think about this. You made a few points on this before, but for a biologist, one of the most exciting things that happens is when you realize you're in the midst of a paradigm shift. And often that paradigm shift isn't fully recognized by the rest of the scientific community for a few years. Again, this mismatch between rate of advance in computer science and in biology. So the data that's being used by our agents, I assume, is dominated by volume and not the most recent history. So how do you compensate for that?
Yeah, so it's a great question. And I think one of the really exciting opportunities for these AI scientist agents, and also an area that you and other people here have been doing great work on, is really having the AI and agents to help us to tell us what they don't know, right? And then having them to help us to design new experiments that can best fill in both our gaps in knowledge, but also the AI's gap in knowledge. Right. So one challenge of that is that a lot of the current AI systems, especially the language models, they're really optimized to answer questions rather than to ask questions. Right? Like if you look at ChatGPT, like the way it's trained is that you ask it something and then sort of like Google and it will give you an answer. But really we want to flip this around, right, and say, okay, so have the AI tell us, okay, so here's everything that it knows about a particular area of biology, and then say, okay, what it thinks is sort of the gap in its knowledge, and what will be the most informative experiment, right? So that if we collect that data, that would be the most illuminating for the AI's brain, also for us.
Yeah, yeah, and I love how you put an AI critic agent into these experiments. What was your inspiration for doing that?
Well, I think in some of our own group meetings, right? So we find it is often quite useful to have some contrarian voice, right? If people are too agreeable and everybody sort of goes in the same direction, then it's okay. And but it's often like we want to have some more debates, right? So friendly, but like really people should argue and have debates and go deep into a particular topic, right? And we thought, okay, so one way to sort of spark these debates among AI is to explicitly have this kind of critic agent who tries to be this contrarian, right? And then try to say, okay, like, why wouldn't this work, right? And then you have to convince that critic, right? And in the process of doing that, I think that also sharpens and robustifies a lot of the reasoning process.
You know, you're here at an institution that has made, I think, profound contributions through understanding mechanism. Yeah. And in my class, I tried to teach you about the importance of mechanism, but most of the large language models we and others are using have this interpretability problem with mechanism. And how do you see, moving from prediction to understanding mechanism? I think you started to address it in some of these cases, but I'd like to hear a little bit more about that. I think that is the limitation in 2025 for me.
Yeah, I think that's really interesting. I would actually say that the large language models are intrinsically much more interpretable than other kinds of, let's say, deep learning systems. Like, let's say 2 years ago, if I built like a deep learning model, like a neural network to predict something, right, to predict changes in gene expression from variants and other things, right? So then there's like a big problem of how do I really figure out what is the logic of that deep learning network. But now if I have like a language model-based agent, because it actually speaks human language, speaks English, right? So in some way, it could just directly explain to me its reasoning process, right, through human language. Of course, there's still the question around, can you actually trust its reasoning process, right? It's the explanation that it provides, is that actually how it really works, right? So there's still that gap of verifiability that people and some of my students are working on. But at least I think that reduces a lot of interpretation barriers, because the fact that these models can communicate to each other and to us through natural language. And we see some of that in the virtual lab as well, is that when the agents communicate, everything happens through English or through human language. Right? So all of their meetings is in human language, and then we can actually look over their shoulder and look over their transcripts and get a sense of why did they decide to design nanobodies rather than antibodies. Right? They provide some rationales. And why did they decide to use AlphaFold rather than some other tool? Right? They also provide rationales. And I think that actually makes their reasoning much more transparent.
Yeah. You know, university systems have traditionally been quite siloed. They're departments, and they have specific names, and then you have domain experts within those departments. And we've all known for a long time that collaboration that's interdisciplinary is highly productive. And you're an example of this. Where do you see the cultural or structural barriers that need to change so that we can provide an educational environment to optimize these kinds of collaborations between biology and computer science?
Yeah, so it's a great question. I was a computer scientist and didn't know very much about biology until I took your class, and so it's great that you actually allow computer scientists to come into these classes. And I think that actually ends up being very helpful. And in a lot of our experiences, right, we love to do interdisciplinary research, but often there's a couple of big bottlenecks to that. Right? One is that it's hard to find time from that experts. Right? If I want to, I would love to, for example, have a collaboration with a pathology's experts. Right? But it might take me like 3 weeks just to set up a meeting to get a half an hour on their calendar to figure out what's going on. Right? So that's not great for collaboration. And also maybe they have a bunch of concepts and contacts that I don't have, right? So that makes some communication also a little bit harder. But I do think that this is also an area where AI could potentially help us with, right? Like for example, as part of Virtual Lab, you can actually train a pathologist agent, right? And now instead of waiting for 3 weeks to set up a half-an-hour meeting with a pathology colleagues that are very busy, you can actually just have instantaneous, like 24/7 collaboration with this pathology agent, at least to get started, right? And that can maybe help you get some initial results, initial data to facilitate that interdisciplinary collaboration. And then you can bring in the human experts to, when they're available, to help to further accelerate. Right? So I think one promising area of AI is to basically fill in some of these gaps, missing gaps that we have in our interdisciplinary collaborations, where when the human experts are not available.
I want to go back to an issue that's just important to me personally, and get you to help me. And that is much of what we can do in this space right now is driven by structural models for proteins. Yeah. But about 30% of our proteins are missing in that domain. Because they don't form conventional stable structures. So can you give me an example how we can overcome those gaps in biological data that then would have an impact across many, many domains of interest?
Yeah. I probably should have brought the AI professor here to help me answer some of these questions and maybe have them run like a group meeting on the side while we, to give us the answer.
That would have been great.
Yeah, we should do that next time. One thing that I think is quite promising and interesting is that now we have these quite powerful foundation models, right? Like these evolutionary scale models and AlphaFold style models. Right? And people have been mostly using these models for looking at structures of, predict structures of a subset of proteins, right? But I think there's actually potentially a lot of insights that are hidden in these models that are relevant even for the less structured proteins, right? But we can extract those insights that can help us to characterize them. So just to make this more concrete, right? So in addition to, let's say, you can take AlphaFold, but instead of just asking it to make a prediction, right, you can actually extract different kinds of concepts or embeddings from the model. And some of those concepts corresponds to well-known structures, like well-studied kinds of domains and biophysical properties, right? So that makes sense. But we also found that there are certain concepts you learn from AlphaFold or from ESM that corresponds to less, more disordered parts, right, where more dynamics parts, right, of different proteins that are less well annotated in existing databases, right? But they do actually show up as sort of coherent clusters in this embedding space of this abstract concepts learned by these models. So perhaps I think there's actually already maybe a lot of insights that we can learn from these foundation models if we just ask it the right way, right, and try to extract these additional insights that be relevant for this, these 30%.
Yeah, I agree with you, and I'm just making this a priority for you. Failure modes. Every new technology comes with a risk of overpromising success. And where do you think the field's at most risk of overhyping, overextending for predictions in biology?
Yeah, I think the data, as you mentioned, is still a fundamental barrier, right? Like the trend in the AIs has been really scaling up, previously been scaling up AI, compute and data simultaneously, right? And that's how they're able to get these models to perform quite well is when they scale up, essentially, you want your training data to basically be proportional to the size of these models. That's kind of the scaling law behind these large language models. Right? And that's been quite successful, but that sort of has already been plateauing now because people have run out of a lot of human text data for these models. Right? And in biology, I think there is a lot of data, but often, if you really want to get into like the causal mechanisms, right, so a lot of that kinds of data is still quite sparse, right? There's a lot of observational data you can get from sequencing through RNA-seq and all these things, but that doesn't quite tell you about the mechanisms. And so I think we're still in a pretty early part of the scaling law, especially when it comes to AI for biology, at least for the kinds of like the more, the deeper mechanistic biology that we're interested in.
Yeah. I want to ask you one last question, and I wouldn't ask this if I didn't see this amazingly creative sort of mind you are illustrating here. Some people think that the reason we have a 90% failure rate in moving from a concept to a therapeutic that works in humans is there's just fundamental poor understanding of human biology. And it's not so much an understanding of how to apply chemistry to biology, how to conduct clinical trials. It's really, there's some fundamental gaps. Where do you see, I mean, first of all, would you agree with that? And secondly, where would you see us making really big leaps in the next few years in reducing that billion-dollar cost and moving from a concept to a useful therapeutic?
I would agree with that assessment. I think one opportunity is that there's starting to be larger amounts of what's called like real-world human data. So basically data that you can extract from EHR, but those are also linked to some sort of genomics, right? Let's say in cancer now, people do this foundation medicine test. So they have some of the mutation data, but it's also linked to their treatments and to their outcomes, right? Certainly the larger biobanks in the UK, US, other places also started to collect more complete real-world data of actually real-world populations. And to your point, I think ultimately, like, to fill in that gap, there's nothing that quite beats having just more human data. And I do think that a lot of these real-world cohorts are sort of very good natural experiments, right? In the sense that, when you capture some of their somatic mutations and you also see their treatments, there's a lot of this natural variations in what treatment is given to a patient based on where they live, right? And whether the clinician follows the guideline or not, right? So I think those are really a treasure trove of natural experiments that we can learn about human biology, right, from these large real-world cohorts. I think some of the pharma companies are starting to do that, but not very systematically. So I think there's a huge opportunity there.
James, it's such a pleasure to see you again. Thank you for educating us.
Thank you.